Okay, we now want to discuss the control of striker. Now it is important that if we are going to control striker successfully, we should know the life cycle. Now we can interfere with the striker Asiatic, control of we can interfere with the life cycle of striker probably here by probably limiting soil moisture if we can, or here we can change varieties, find varieties which do not support the germination of striker, although the conditions could be there for preconditioning. Then we put a variety which doesn't produce what these parasite seeds need. Or we can cause problems for the striker here by finding varieties which do not allow penetration to take place. This is the relationship of grain weight striker, crop grain weight versus total dry weight of striker. If you increase the density of striker, this diagram gives you an exponential decay curve. What it means is that you only need a few plants of striker to cause big yield reduction. So if you see a few plants of striker in a field, you should take action. Otherwise, you can have serious problems. So the key points to know is that striker damages the crop before emergence. This can limit our mechanical methods of weed control. And also, a small amount of striker in a field can cause serious damage. So, even if we see few plants, we may say take action. But with the other ordinary weeds, there is always a threshold where we say this density may not affect yield, but with the striker asiatica or with the parasitic weeds, even if there are few plants, weed control measures have to be taken urgently. You may want to read this reference which summarizes weed control measures or striker control measures and their problems. Control the striker conodrum in pesticide science by Heaney. Or you may want to read this current progress in striker management, which means there are challenges when it comes to control methods. Now let us look at the host parasite relationships. Now a host can be susceptible. What it does is that It can have high striker emergence and low grain yields. If the host is tolerant, it may have high striker emergence but also high grain yields. Partial resistance, you have less striker emergence and slight grain yield reduction or no grain yield reduction. Probably these are the sort of varieties which we need. Resistance or immunity, no striker emergence, high grain yield. If we can get such a variety, that could be very useful. So one of the control methods which we can use is that we can look for resistant crop genotypes, because you need yield first. There have been some resistant sorghum varieties or cultivars which we have identified in this country. SAR29, SAR33, SAR35, SAR37, and SAR16. These can actually be used to make some crosses which become resistant. For example, SV1 was crossed to SAR29. It was found that recessive gene controlled low stimulant production, and it was known as the LGS gene. So production of stimulants depend on major genes. So it is possible to solve this problem through breeding. Here there's a paper where we tried to determine varieties with the low stimulant production.
Dok makamute hatisi kunzwa.
Hello?
Makaisa pamute.
What is the problem? Now, resistant sorghum cultivars, we have sorghum land races. Kasvikisire, katandadzara, mutode, nyamudzi, muteruwane, ndigano. These, I think these varieties, I think these land races are tolerant to striker.
Hapana chiri kunzwika Dok.
Hapana sound. Okay. What about now?
Hatisati takunzwa. I think mabaya mute.
Oh, okay, let me. Oh, yes, you are right. Thanks. Can you hear me now?
Yes.
Thanks very much. Saka zvatangira papi?
Pamaenda ndopata kumusira kungu pamazodzoka mamamuta.
Pandatanga Parasitic weeds, mamuchizvinzwa. All right. So can we continue?
Yes.
Thank you very much. Thanks. Okay. These are some of the papers. I'm not going into details with them, but you can always follow them up so that you understand about resistance. There are also tolerant maize cultivars like the CML 395 and R201. R201 is an old hybrid. I think it was specifically bred for drought tolerance. And at some stage I said drought is associated with abscisic acid as well as striga is also associated with the abscisic acid because they control stomata closure and opening. This is how we actually identified it and tried to screen it for tolerance. And indeed we found that it was tolerant or resistant. But there are other cultivars. This R201, I think, I don't think it's popular with farmers or with the people who are selling this variety. At one time, Ada was producing it. I don't know whether they are still producing this variety. Now in Tanzania, Mbwaga Masawi in 2001 talked about these varieties or genotypes with resistance. I think these were specifically bred for resistance. Probably the question is, do we have striga resistance in maize? Yes, we have got sources of striga resistance in wild relatives of maize. For example, in Zea diploperennis and in Tripsacum dactyloides. These are grass-like plants. Now, at IITA, they were able to develop a line which was resistant to striga species, which they named ZD05. Now, this was a cross between Zea diploperennis and tropical germplasm. It indeed reduced the striga agents. This is Zea diploperennis, a perennial teosinde or wild maize. This is Tripsacum dactyloides, which resist striga asiatica attachment. Now, if we look closely at the ZD05, it can avoid striga because of its less branched root architecture, but this is not a very strong trait for resistance. But the second one, where it can actually resist attachments, this is very important. It also doesn't support growth after attachment. So it interferes with the life cycle of striker. This is also an important trait. You may have a look at that paper. There's also a paper where we looked at the reaction of new quality protein maize genotypes to Striga asiatica. This work was done in Gokwe. Again, another paper which you may want to have a look. Here we have germplasm bred for resistance to Striga asiatica. This was done in this country. We did this with an MPhil student, Vimbai Huayo. And of course, we have Professor Gasura leading this project. Again, more papers on breeding for Striga resistance. So here at the University of Zimbabwe, some work was actually done in the maize, so that we now have some varieties of maize which may be sold under VN Seeds. There's a VN Seed company within our department or within the university, which may start selling some of the maize varieties. We could have maize varieties which could resist Striga asiatica. So they are now products. Another paper on Striga asiatica. This is about breeding. We had an MSc student, Rwafa, who did also a project. Rwafa was an MSc student who was involved in much of this breeding for Striga resistance. He is working at ADA. This is Striga emergence in polythene bags, which we used for screening. So these are the bags for screening. This is the evaluation of maize hybrids in Gokwe communal area. This is a tolerant hybrid in Gokwe. You can see good crop. Striga, this is now maize plant which is, very susceptible. You can see this maize cob, the good maize cobs. This is under field evaluation of maize hybrids in Gokwe. Here you cannot see Professor Gasura, but you can see his hands holding this cob. Again, there's a paper so many papers about striker resistance in maize done by our department. If you have time, you can read these stories. We can move on to, so what we are saying is that we now have some hybrids in the country, although they have not yet reached the farmers. But very soon they will be released, which can resist Striga asiatica. Probably for the first time we have these varieties. Now we move on to tobacco tolerance to Striga gesneroids. We have T66, which was found to be tolerant to Striga gesneroids, followed by T61 and KRK26. This was reported by Koga. Well, this is a very susceptible tobacco variety infected with Striga gesneroids. Here we had a student, Chapman Koga, who did a PhD or a DPhil on genetic characterization of Striga gesneroids and its control strategies in tobacco. He was our student. After finishing, he migrated to Australia, and that is where he is working now. We thought we could retain him, but he found better things to do. This is a paper by Koga, if you want to read it. Screening of tobacco genotypes for tolerance resistance to Striga gesneroids infection in Zimbabwe. Again, same paper by Koga. Now we move on to mechanical methods. Hand hoeing and pulling to prevent seed addition. We may recommend that farmers should try to minimize flowering and the production of seeds. And this can be done in February. Striga plants may be burned. But remember, this is going to take a very long time before the crop can benefit. Remember, the seed can last for 20 years. So it's a long-term program. Herbicides, we do have old herbicides like 2,4-D, MCPA, which were used by the then commercial farmers, and they succeeded probably in suppressing parasitic weeds. But however, when we apply these post-emergence herbicides, the damage will have occurred. But then there's this dicamba, which can be applied at 4 weeks after crop emergence. It can also suppress Striga. Dicamba, we have it in Zimbabwe. All of these herbicides, we have them. Now, one of the most effective ways of suppressing Striga is to use imazapa-resistant maize or IARA maize. This IARA maize was developed by CIMIT. So the control strategy has got two components. Number one, a herbicide-resistant maize seed, And number 2, you have the herbicide, which is imazapa, a systemic imidazole herbicide. So the herbicide-resistant maize is coated with low doses of herbicide, about 30 grams imazapa per hectare, to control striga. I may say at this stage that in West Africa, in East Africa, they've now developed maize which is resistant to this herbicide, and they just dress the seed with the herbicide. So farmers can plant seed which has already been treated with the herbicide. And after planting the seed in striga infested areas, their maize can actually yield better. Now, if you want more information on this, you can read this paper if you are interested. But the message is that we now have maize which can be treated with the imazapa. It's already carrying the herbicide when the farmer buys the seed. Now, there's also another paper here on imazapa. Now, on this paper, Zimbabwe also contributed in this study, where you have Lawrence Jassy as one of the authors of this paper. So there were some sites in Zimbabwe where this work was done. Now, crop rotations, we can use trap crops or fossils to in rotations. For example, we can use bambara nuts. Now, BN Dagutang and TSVU 1033, BN Chizanga, BGN 113, and Chipata 1 were bambara nuts which were found to produce a lot of striga stimulants. They could cause a lot of germination of striga, but yet they were not attacked. We also have cowpeas, which produce a lot of stimulants, yet they are not, yet they do not support striga. So what we are saying is that in crop rotations, you need to use such materials so that they remove or kill striga. When you come up with your maize after these legumes, the challenge from the parasites will be reduced. So there's a paper to that effect if you want to read it. Catch cropping, that is growing a susceptible host and plowing it under. So what you are saying is that you allow farmers to grow crops. At flowering, you say destroy that crop in order to reduce the striga seed bank. But this may not be attractive to farmers because they don't want to waste their time. They can use the Sudan grass. Probably this can be done to abandon the lands But I don't think farmers will want to do this. They want to grow something and harvest. Not to say grow this crop, destroy it, maybe after next year you get better yields. I don't think farmers have that patience. Intercropping, we can intercrop maize and cowpeas, which can be grown in the same field to reduce striker emergence, and this can increase grain yields. There's an MPhil student, Kasembe, who did an experiment as far back as 1998. Sorry, coming. Then there is Khan et al., who also did an intercropping with maize, with the Desmodium cynatum, which greatly reduced Striga montica in Kenya. So what we are saying is that intercropping maize with legumes like cowpeas. In Kenya, they did intercropping with the Desmodium cynatum, which reduced the striga. So this could be beneficial. So here are some of the papers on this intercropping. If you have got time, you can read them. Now, this was reported by Kasembe. Just on these two tables, you can see that it didn't matter which cowpea, as long as you mixed the maize and the cowpea, the yields were always increased. So here we've got sorrel maize. If you compare sorrel maize with intercropping, you find that there's less yields in sorrel maize. And this is because of the effects of intercropping on the parasite. So you can explore this on this paper if you want. There's this climate smart push-pull, which I want to explain. Here we've got a problem where we have a parasite. This is sorghum. Then here we've got bracharia grass. So you have got a field of sorghum which is surrounded by bracharia grass. Right, so we have got Desmodium as the intercropping with the sorghum. Here we have got actually two problems. We have got the stem borer, which can destroy sorghum. We have got the parasitic weed, which can also destroy the sorghum. So what is going to happen is that the Desmodium will produce chemicals which will stop the striga from attacking the sorghum. It also produces some chemicals which drive away the stem borers. These are the pushing chemicals. That's why we have the push word here. Then the bracharia also produces some chemicals which attract the stem borer. So we have the stem, the bracharia pulling the stem borer, and the desmodium pushing out the stem borer from the sorghum field. So we have the push-pull, or the push-pull system, which takes care of two pests. It was developed in Kenya by Isipe. Here we've got maize and napier grass. We've got maize surrounded by napier grass, but maize intercropped with desmodium. Now this desmodium is driving away the stem borers from the maize crop, and it is also producing chemicals which affect the striga. Probably at this juncture, I may say that there's a student here at UZ doing a DPhil more or less along these lines, where he has sites in Murewa, where he has desmodium mixed with maize, as well as napier surrounding maize plots. And he is looking at the impact on other pests in the maize fields. Nitrogen management or fertilizer. For a long time, it has been known that poor soils are associated with the striga. So nitrogen can reduce the impact of striga on cereals, but the effects of nitrogen depend on crop cultivars. Now, some scientists like Agbor in 1991 recommended the use of 120 kgs of nitrogen just in order to induce striga tolerance in maize. But this could be too high. So here in Zimbabwe, levels like 30 kgs were tried on CML 395 and R201, and were also found to be effective. In tobacco, 50 kgs per hectare were tried and found to be effective in reducing striga gaziniroids. So addition of nitrogen can actually reduce the impact of striga. So what are the mechanisms? How does nitrogen affect striga? Nitrogen could decrease the number of germinating striga seeds, as reported by Peters in 1990, or there can be low stimulant production or low root exudate production, as reported by these authors. There can also be inhibition of striga attachment process on the host root. The other issue is that nitrogen can stimulate photosynthetic activity of both parasite and the host. So increasing nitrogen or making sure that there's adequate nitrogen in the soil may reduce the impact of the parasite. Another element is phosphate. For example, diammonium hydrogen phosphate applied to sorghum reduced the production of strigalactons, sogomol, and 5-deoxystrigol were reduced. Striga emergence was also reduced. If you want more information, you may read this reference indicated. Here we had an MSc student who tried to use phosphorus to reduce striga attachment. On this graph, you can see if you increase phosphorus, you reduced attachment. He used the agar gel technique where you have maize roots growing through the infested areas of striga, and in these petri dishes phosphorus was varied. He had 6 maize genotypes, and all the 6 maize genotypes had a tendency to show the decreased striga asiatica germination or emergence. So it didn't matter which variety. Timely planting. In this country, we found that if we planted maize early, we could reduce, we could increase grain yields, but the number of striga infestation was not reduced. So we still recommend that farmers should plant early in the striga endemic areas so that their maize may tolerate the striga infection. But of course, they may need to come in with the supportive hand weeding or pulling to remove the striga plants. If you are interested, I may supply you with this paper. So let us look at the biological control. In other areas, they have used insects like Simincronix, which control the striga emontica. Others use the fusarium isolates. One interesting issue is the avascular mycorrhiza proposed by Ledzemo et al. In 2007. Now, when you have avascular mycorrhiza, the cereal will provide the fungus. Avascular mycorrhiza is a fungus. So it will get the carbon from the cereal, and in turn it provides phosphorus. And this availability of phosphorus will end up suppressing strigalactons. And these strigalactons, which are suppressed, will ensure that the host is free from the parasite. So the avascular mycorrhiza colonize the roots of the host, which are also the target of the parasite. Once the avascular mycorrhiza is present, it creates an environment which is not attractive to striga infection. You can read some papers about it if you are interested. about how a bascular mycorrhiza can be used to control striga. Yeah, we have got these other parasites, Electra vogeri and Striga gesneroids. They have a similar life cycle to that of Striga asiatica. The control is also challenging, although we can use hand hoeing and pulling, but there are limitations. You may be interested to know that cowpea B301 was found to be resistant to both Electra vogeri and Striga gesneroids. And also the addition of phosphorus was found to, at 45 kg per hectare, was found to reduce the impact of Electra vogeri on cowpeas. This was reported by Mugabe in 1983. So I think the research should focus on identifying cowpeas and bambara nut which are possibly resistant or tolerant to these parasites. Here there's a paper by Rabaguzibwa on cowpea and Electra vogeri. Another paper on screening genotypes. There's a BSc student who also tried to screen a lot of genotypes to which we direct Electra. So you can see the effects of Electra on cowpeas. You can see the leaves are dying. This is Electra in pots. Now, Cascuta campestris, it looks like spaghetti. This is actually attached to the shoot of the host. Now, this parasitic weed, Cascuta campestris, Doda, is a problem in Lucerne. However, it has been parasitizing tomatoes, carrots in the US, even at grasslands at Horticultural Research Center. It was also found to be parasitizing horticultural crops. The shoot parasite has got little chlorophyll. The stems are yellow to orange. And like I said, it looks like a spaghetti. So let us briefly look at the life cycle. We start with the seeds, which are very hard and can be dormant and survive up to 50 years. Now, with the seeds, these seeds do not require a germination stimulant, unlike the other parasites. Once there is moisture, if we move to this stage, and you have got temperatures 30 to 33 degrees Celsius, you have a seed germination. Now, after germination, the seedling will look for the host. You have an elongated stem. Now, this stem will start to search. It actually moves. It grows very fast. It moves in a circular fashion or anti-clockwise fashion, which is known as circumnutation. Once it gets hold of its host, it grows right around the stems. Whilst it is doing so, it forms attachments. You will have ostoria at certain points, which enables this parasite to get carbohydrates from the host. So it uses cutin to form attachment and also cytokinins. After a while, it produces flowers, and these flowers will give us seeds. So again, as I have said, you can start with hard seeds, which are dormant, which can survive up to 50 years. Of course, the temperature range 15 to 39 degrees Celsius could facilitate the germination. But the key point, no requirement for germination stimulant. This is a shoot parasite, not a root parasite. It emerges and forms a single root, and the shoot will swing in a circular motion or anti-clockwise motion about once every hour, which is known as circumnutation. It coils around the host, forms ostoria. It also forms cutiny for bonding. Then ostoria development starts, and it also requires cytokinins. Now the parasite can reduce the growth of the host. It depends on the water, mineral salts, and the carbon. It can also affect photosynthesis, which becomes inefficient. Now on the control measures, you can see with the shoot parasites, it's very challenging. If you want to control the parasite, you might as well destroy your crop. So hand pulling and pruning of infected plants, you might have to lose a few plants. You may burn the parasite. If it is lucerne, mowing can be done to remove the parasite. There's need to identify lucerne which is resistant and tolerant to cascuta. Of course, some herbicides like pendimethalin and trifluralin have been used. Some have tried to use gramoxone and glyphosate at low doses, but sometimes you end up burning your crop. Now, in conclusion, we may say that parasitic weeds are some of the most important weeds in Zimbabwe, and there is urgent need to look for methods of controlling them. We may use tolerant or resistant genotypes, probably as a focal point, so that we can develop integrated control systems, because no single method is likely to be useful to control the parasitic weeds. So this is the summary of parasitic weeds. Root parasites, we've got Striga asiatica, Striga forbesii, strike against neroids, electravogeri, ramphicapa fistulosa. Then we've got one shoot parasite which is important in agriculture, Caescuta campestris. This is the life cycle. And these are the effects in summary. We talked about things like, wilting, standing, reduce the stem biomass, reduce the photosynthetic rates, etc. So this is a summary of striker effects. And again, this is a summary of the control methods, where we start with tolerant crops or resistance, hand weeding, etc. So you can go through this summary. I will not go through it again. So this is it. Hello.
Hi, Doc, we're around.
Yes, now we are through with the control of parasitic weeds. Any questions? On parasitic weeds? Otherwise, I would like to introduce you to chemical weed control, a very short session, so that when Dr. Rugare comes in, I will have at least introduced you to the herbicides. So let me stop streaming. So from now on, we will be talking about weed control measures. And obviously herbicides play a big role in weed control. So I just want to briefly introduce the chemical weed control. I want to make these slides move. Okay, the question we may ask is, what is a herbicide? Simply, it's simply a chemical used to control weeds. You can have selective herbicide. That is the herbicide which can kill weeds leaving the crop unharmed. For example, atrazine in maize. We also have non-selective herbicides which can kill a plant or plants which it comes into contact with, or which could be glyphosate. So that chemical which doesn't discriminate in terms of killing is non-selective. So we have herbicides which are packed in containers, and they have some instructions. If we look at the world map, we may find that in the northern hemisphere, where we have these developed countries, this is where most of the herbicides are used, as well as Australia, South America. There's also South Africa, China, a bit of India, Egypt, and well, some countries in Africa use them, especially in large-scale farming areas. But smallholder farmers hardly use these products. This is just a brief history of herbicide technology. In 1896, copper sulfate was used for broadleaf weed control. By 1912, an attempt to use sulfuric acid, but it was not until 1944 where real herbicides, as we know them today, were developed. And the first herbicide to be developed was 2,4-D, 2,4-dichlorophenoxy acetic acid. This could actually kill weeds selectively in cereals. In the 1950s, we had the triazine herbicides being introduced, and the 2,4-D was also being used in Zimbabwe by that time. In 1965, we had the weed research team establishment at Henderson Research Station, which was supposed to research on weed technology improvement, specifically focusing on the use of herbicides. By 1974, glyphosate was introduced as non-selective herbicide. So when applying herbicides, we need sprayers. It can be also tractor boom sprayer or knapsack sprayer. So you need something to apply the herbicide. You need the knapsack or tractor boom sprayer. Of course, nowadays, I mean, you can have the use of a drone or an aircraft applying herbicides. So what are the advantages of herbicide technology? They can eliminate early crop weed competition, leading to higher yields. You know, when we're talking about the critical period of weed control, you can actually reduce competition through application of the herbicides. You can reduce time spent weeding. Labor is released to do other jobs. It removes labor bottlenecks which occur from November to January. And there's a possibility of increasing the area which is planted. It can also be used under wet conditions. In fact, under wet conditions, mechanical methods are difficult to use. Now, it can allow this reduced tillage or conservation farming to be implemented. You know, conservation agriculture or Pfumvudza is a climate smart technology which can increase crop yields through timely planting. Its efficiency can be increased by the application of herbicides. Improvement of quality of life in smallholder farming areas. Chivinge reported that women spend 75% of their time weeding in the peak weeding periods from December to February in Zimbabwe. So if we use herbicides, these women can be released from this weeding, because most of the time they have to spend, they have to weed, especially between December and February. We can effectively deal with the difficult to control weeds, e.g. Cyanodon and Cyperus esculentus. However, there are disadvantages. Herbicides are toxic substances. Here, if we talk of LD50, that is the amount of poison which kills half the organisms in a randomly chosen batch of named species, e.g. rats, measured in milligrams poison per kg body weight. So LD50 gives us an indication of how poisonous is this herbicide. Paraquat has got an LD50 of 100. We represent it with a purple triangle. It's very toxic. 2,4-D 300, LD50, red triangle, MCPA 700, amber triangle, glyphosate 4,320. This one is less toxic, a green triangle. So this information should be available to any person using the chemical. So herbicides are poisonous. We can indicate it by this sign. Now, herbicide residues could end up in drinking water. It's a disadvantage. You may need to transport large quantities of water for spraying. Clean water may also not be available. Now, although herbicides have been available, you find that few farmers, especially under smallholder agriculture, use these herbicides. The question is why herbicides are not used widely by smallholder farmers in Zimbabwe. Chivinge in 1984 gave a figure of 1.5% of farmers who were actually using herbicides. That is smallholder farmers. I don't know whether this figure has changed. Now, some of the reasons why farmers may not be using herbicide technology, according to Chivinge, was that there was lack of awareness. During that time when that survey was done, about 99% of the farmers were not aware of herbicide technology. But this could have changed by now. Probably more farmers are aware of the herbicide technology. A few extension staff carry out herbicide demonstrations. Probably this is too risky. If you make a mistake, you can destroy the farmer's crop. There are also challenges with the sprayer calibration, which involves a lot of volume measurements and doing calculations. Making mistakes could be costly. The instructions are always in English, and probably farmers may need a different language. Also, cash is not available. So cash or capital, there's limited cash to purchase inputs at the right time. Farmers who sell their maize to GNB are sometimes not paid in cash. Sometimes farmers who grow crops at a loss, e.g. cotton or tobacco. If farmers have capital, then they can be in a position to buy these chemicals. But sometimes this is a serious problem. No credit facilities. Banks are not financing crop budgets. Lack of collateral with smallholder farmers or immovable property. Banks are not using land as collateral because it's risky. Low rainfall, climate change. Farmers may consider herbicide technology to be risky. Frequent droughts and unreliable rainfall leading to frequent crop failures. If there's a serious crop failure and the farmer has used the herbicide like this previous season, then the farmer is not likely to use it again. poor infrastructure and support systems. There may not be regular supply or regular and reliable supply of agricultural inputs, especially in rural areas. However, sprayers and insecticides sometimes can be found in some local shops, with the exception of herbicides. If the herbicides are there, they are limited. There's very little choice. Then packaging, sometimes herbicides are sold in large packs of 5 liters. Most farmers probably need 3 liters or less. And if you have big quantities, then the farmer is not able to find the capital. So this is herbicide packaging. But probably the packaging could have changed now. Instead of having just 5 liters, maybe 1 liter containers are now available. Now there was a survey which was carried out by Makangani Setal in Chiota, Narira, and Cholocho, which were used as demonstration sites for herbicides, just to find out from farmers what were the issues surrounding the herbicide technology. Now, the technical issues which were of concern to farmers was the time of herbicide application, especially use of herbicides under dry planting. Sometimes farmers, would plant under dry conditions. They wanted to know if they could apply herbicides. Now, repeated use of herbicides in the same season and between seasons in the same field, it was also an issue. The amount of herbicide applied per hectare, herbicide residual effects, how long does the herbicide last being active? What about intercropping and crop rotations? Can these herbicides affect these systems? Also, technical issues. The issue of toxicity versus efficacy. Does it mean the most toxic herbicide is the most efficacious herbicide? Is there a possibility of replanting fields where herbicides have been used in case of poor germination? And also range of herbicide formulation, liquids, granules, etc. Managerial issues. Some of the extension messages around herbicides were not very clear to the farmers. Language on labels difficult to understand. Availability of herbicides. Sometimes they don't find herbicides in shops. Cost-related issues. Cost of the herbicide versus traditional weed control methods, which is cheaper. Some farmers may think maybe hiring labor is cheaper. Cost of knapsack sprayers. During that time, maybe the knapsack sprayers were very expensive. And environmental issues. contamination of food by herbicides. Let's say if you use the herbicide, is it safe to eat the crop? What about the stover, which is eaten by animals? Is it safe if we use herbicides? So this is it on herbicide, or the herbicides introduction to herbicides. Let me come back. Are you still in?
Yes, still.
I can see we are now, there are now four of you. Right, so this is it on the introduction of herbicides. I don't know, from your experiences, do you think smallholder farmers are now using herbicides? Or they just use hand weeding?
Most use hand weeding because their plots are small and the family forms the labor source for the land. So it's cost effective for them to use their hand weeding.
Oh, okay. What about the prices of herbicides? Do you think they are affordable for farmers? Especially the cost of Stella Star. Do you know what is the price of Stella Star?
Yeah, it's costly, about $70 a liter.
Or $70 a liter?
Yeah, $70 a liter. So it's so costly that smallholder farmers cannot buy them.
Yeah, even lecturers may not be able to buy them. If they are $70 a liter.
Oh, yeah. And also there's this other pre-emergent website, the Integrate. It also costs nearly $60 a liter. So yeah, of which those are the herbicides that are so effective, but they are costly manje. Ukuthi smallholder farmers, they cannot buy them, so they end up vakutongo takura ivo personally, and some vachiri kungofunga kuti ma-herbicides anoda sei, especially kuno ku this side kwandiri kuno, vanototaura kuti ndiri kunonzi kumiddle suburb kuchipangayo.
Okay.
Yeah, they are saying kuti if you apply ukaisa monzi musugar beans mame, they are saying kuti ukashandisa beans dzinenge dzakasakurwa nevanhu ne-beans dzaiiswa herbicide, if you try to pay yield inobuda to be different, kuti kuti ma-herbicide akaita sana bendazone akaita sana chii, they have an effect yaanawo pakuya, ndo kutaura kwanenge vachiita kuti
Probably
anoita
that's the phytotoxic effect, isn't it? But the legume will always grow out of that phytotoxic effect.
Yeah. So that with that effect manje, some farmers they are now thinking kuti since vanenge vachitohauma ma-acreages anenge ari madiki, nearly 10-20 hectares, so they are now considering kuti haya vanhu and using my-herbicide.
Okay.
Yeah.
Anyway, this is it. Anyway, just find some issues on the smallholder farmers. But I think on other farms, herbicides could make a big difference. So tomorrow I think you will have Dr. Rugare talking about these herbicides. But as I've said, I'll find a slot where I'll quickly explain about weed control thresholds, which may not be probably clear on the area slides. I'll see when I can do that, or I'll just post a pre-recorded lecture so that you go through it at some point. It will be a short one, just to explain some issues on those graphs which I was talking about during the first lectures. Otherwise, I'm going to stop here. Any other issue?
Nanda kufunza talk in relation to the assignment. Tiri kusubmita on which platform?
Munondisendera by email.
Saka tombrozesera your email address mu group.
All right. Or you can physically hand in if you want.
Yanga iri Juna saka tatisiri pa campus.
Okay. Yeah, just I'll put an email on the Papa group.
Okay, Tom,
so
thank
you.
you.
Can send. Otherwise, Mama Pete.
Hatisati.
All right, Kanama Pete just send that to the email.
Okay.
Yeah. Probably pani muni baba assignment amakapo.
Tine mama chakapona total gun.
Yeah, I'm going to give you some, maybe one or two.
Okay.
Yeah, pani zvatakabva nhasi, maybe one or two.
Yeah.
But for what I am sending you, I just want you to try and answer the questions. Musazoise my references.
All right, talk.
Because I want you to imagine you are writing an exam question and you are answering it. That is the purpose. To practice so that you can be able to answer these questions. Saka mukakurumidza kundipa ndokupaiwo feedback nekukurumidza so that munopinda muexam muri prepared.
Okay, talk.
Okay, so toenda zvedu
pakanaka apo tiri kuti apabata because the assignment yacho it's a bit confusing though, but tichangouitawo zvatinenge tichangonzwisisawo then mozotigadzisa from there. I think matozopindawo muexam tinenge
those are typical exam questions handitika.
yeah.
Pamwe panogona kunge paine nequestion yakatodaro.
Yeah, that's true though. Saka tikatokasa tasenda then maybe moyasoticorrecta kuti tofana kuzoanswer sei tasangana at same question.
Ndoari kuita mukandipa manhingi ndikamaka ndendichiita ndokupai nhingi ndendichipa direction kuti ungadai waita so ndokunge waita error.
No, that's fine. Thank you.
Yeah. Okay. Regai ndichiita zvimwe.
Okay, Joe. Matabasa. Kuswera zvakanaka.
All right, thanks.